I'm very excited today to sit down with a panel of experts on VHHs or nanobodies. I'm joined by Dr. Serge Muyldermans, Dr. Katie Wolfe, and Dr. Sam Sugerman, who are all have years of experience with VHHs. They're an exciting modality of antibody, but over the years, we found that a lot of new scientists in the field are lacking in practical knowledge about how to use them, how to humanize them, how to develop them into reagents and therapeutics. And a lot of that knowledge is siloed and not necessarily taught at every university that the kids are attending these days. So over the past few months, we've just been collecting, soliciting questions from the community, scientists in the industry, and so would like to put them to this expert panel. And so with the breadth of kind of expertise and experience that you have, we've seen kind of an increase in the use of VHHs in the clinic as well as reagents. So I guess put to each of you, where do you think the biggest transformation has occurred due to the adoption of VHHs? In other words, where do you think they've had the biggest impact? Serge, would you like to go first?
Yes, okay, fine. Well, the impact you can look at it in several ways. So if you see in the number of papers that have been published and the amount of work that has been done, then of course the greatest impact for VHHs is still in the fundamental sciences. So to determine the structure of the antigen, of its target, or to track the presence of the target within cells, or to unravel the enzyme mechanism. The enzyme is then the target as well, the antigen, or to see with which other enzymes or partners a particular target is interacting. So that's the biggest thing. But of course, economically, that's less important compared to therapy. In therapy, that's the biggest financially important aspect of nanobodies. But we have only 4 products that are approved for the moment, and not worldwide. Yes, but for example, Nanosora is only in Japan. Yes, but that of course brings in a lot of money for the company, so for the pharmaceutical industry. And then we also have the diagnostics, the tools or applications. It's mainly in academic that people are looking for new developments, new technologies to whether it's an ELISA or biosensors or laminar flow assay, you name it. It's to be cheap and easy and more sensitive, etc. But the problem is that it has not been commercialized. I thought this would be the first step to commercialize, and in the end, it's the last one that gets commercialized. There is one exception in diagnostics, and that's the non-invasive imaging, in vivo imaging. For example, for breast cancer, so you can label your nanobodies, inject it intravenously, and within 10 minutes you can take a scan of the patient to see where lesions are or where metastases are located. But the problem is it is not commercialized. It's in phase 2 as far as I know, but it's not commercialized yet.
Yeah, I totally agree with all of those points. I think I have very similar notes. The one additional point I think is that a lot of this work that's ongoing, they really utilize the unique format of these single chain antibodies since they're so small. People are starting to, or have been treating these like building blocks or beads on a string. And so like with regards to therapies, for, I don't know, a few years, all of the engineering meetings I've been going to, I see all these amazing talks about how people are utilizing these to make multi-specific formats without having to do any light chain pairing. And it makes it easy and cheaper to make because you don't have to make it in a eukaryotic system necessarily when you're producing it for manufacturing scale. So you can make a bispecific or a trispecific where you're bridging 2 different cell types for cancer treatment or diagnostics. But you can also add another modality to it to make it longer lived in the serum, so that it doesn't get turned over so quickly. So it's really kind of the sky's the limit on the formats I've seen people put together. They'll have like an FC region, and they'll have nanobody here, nanobody there, nanobody there, and it's really amazing to see how they get it to work.
Totally agree. I think the tunability that Katie mentioned is a huge point. We can extend serum times, but then we can also get really rapid clearance. So if we are doing non-invasive imaging, especially with radio tracers, then there's rapid clearance, and you can actually load up a little bit higher. There's a decent amount of PK data. The other thing is that these are very tunable, and I think that for the bispecifics and multispecifics, that's really important on sort of a cytotoxicity level. One of the things that sort of early anti-CD3 arms realized was that there was a high cytotoxicity if you're not tuning the affinity precisely. And so I think VHH has a much wider range of interaction, and that's very tunable, and you can pull that out pretty easily and quickly because, again, sort of rapid discovery, no light chain pairing, and they move well into recombinant forms.
So then what I'm hearing is lots of opportunity, but then maybe underwhelmed with kind of the performance, given that they've been around for 30 years since your paper came out, Serge. So I guess last week when I talked to Brian Chait, that was one thing that he brought up was that we have all this promise. They penetrate, they're modular, they're stable, they can be tuned in all these different ways. Why aren't we seeing more from nanobodies? I guess, do you have any thoughts on that, or do you agree with that?
Well, I agree, but there are reasons for it. Huh? So the thing is that Hamers, he took a patent on the use of all fragments from heavy chain antibodies. And so we have been blocking, at least for commercialization, we have been blocking the field for 20 years. Yes, so you were not allowed to use them or to commercialize them until 2014. And that, of course, gave us a big advantage because we could work with it, we could do whatever we like, so we could found ablings with our patents, based on our patents, and so also on our patents when they became available, there was a lot of opposition. So when that happened, we knew that it was a good thing because it was worth something. We knew we were blocking people, and and that helped, of course, to create or to found a spin-off company. But now you can see it easily if you just go on my citation index. You see after 2014, oops, it jumped tremendously. So I know everyone is doing it. In nearly every university, people are working with VHHs. Well, in 2000, it was only our university that was working with VHHs. And when I went to conferences, they said, are you still working on it? And so, but now it has been exploding the field.
Yeah, I totally agree to all of those points. I think also you see it takes time to get things through the clinic, apart from COVID. So it's this like you see, I think, what did you say? 3 or 4 products that are commercial, I think. But if you look at the clinical trials that are in progress, it's a lot.
Yeah.
So I really think that in the next couple of years, we'll see, just like the publications, when you look, you see this peak. I think in the next couple of years, you'll see the commercialized therapeutics start to increase. And hopefully once more get out there, it's kind of a snowball effect where more will come because people will have more confidence in the ones that are already being used. Because it's a new technology. So, and you're putting it in a person. So it's some hesitancy on the uptick too, I think.
It's correct what you say. So biochemists or scientists, they are very conservative. If you're working with a tool, you keep on using that tool. You don't want to switch. Or if you're used to an instrument, you don't want to switch your instrument. Yes, you
Right,
keep
exactly.
working on it. And so it's very difficult to convince people that nanobodies are as good as single chain FEs or classical monoclonal antibodies. Well, if you work on it, of course. And that's, well, there we had still a small advantage compared to DARPins. And we have also those artificial scaffolds, DARPins, anticallins, and so antibodies, etc. But those are further away from an antibody. And so doctors, medical doctors, they don't want to use these, but they have no knowledge about it. And so they are not keen on working with it either. And so nanobodies, we can still sell it as an antibody and then say, oh, an antibody, okay, we know.
Definitely. I totally agree.
I think the other thing that we're going to maybe experience here, hopefully, is that there's going to be this coincidence of next-gen therapeutics. So we mentioned bispecifics. CAR T and CAR NKs are the other ones where I think that there may be a huge impact, especially as people are realizing sort of the liabilities of scFvs in some of these assemblies. Things like the poly-GS linker coming into play. And then again, sort of immunologically, I think the hesitancy is valid, right? We want to be conservative here with therapeutics, but my hope is that there's sort of an explosion as we move forward, and there's a coincidence of this new scaffold as well as these sort of new applications that we can really move therapeutics forward pretty rapidly.
Yeah, and if you look at the field of immunotherapy, I mean, that's pretty recent. You know, it depends where you're looking from, but it's not a very long-lived field, immunotherapy. So I think I have a lot of hope that the VHHs in the clinic are gonna be coming soon.
Yeah, and things like nivolumab have been around barely over 10 years, and that was one of the first really impactful solid tumor IOs.
Yeah.
So then kind of looking at the very earliest stages of a new scientist making a decision about starting a new discovery campaign, a lot of times what we hear from people is that they're surprised at how long the llama and alpaca immunizations take. We hear, 8 weeks at a minimum, but typically 10 weeks, and sometimes people immunize for years to get good titers. Is that just kind of this, is there a fundamental limitation, or is that just how we're doing it because that's how we do it?
I don't agree with this statement. We are using actually 5 weeks. We are doing every week an immunization for six immunizations. So you start on day 0 and then until week 5. And then we take the blood. That's most of the time enough. Sometimes for difficult targets that are very low in immunogenetic, then it might be a problem. You need to boost again. But instead of 6, actually those 10 weeks or 12 weeks, it's coming from the hyperimmunization of a rabbit, the old vaccination scheme. Today, 2 weeks, 1 month, and 3 months, yes? But you can do it much shorter. We do it, as I said, it's about 5-6 weeks. And then to make the library, it's 1 week work, and to do the panning, it's another week. So within 2 weeks, from the very first immunization, we have our binders. And then, of course, that's the long procedure. You have 10 binders, 20 binders, whatever. You have to grow them individually, and you have to screen them, and you characterize them to see which one is the best to bind to an important epitope with the highest affinity, which is well expressed, which is stable. And so that's linear work. So it's 1 by 1. But all the rest, it's very easy. And during the immunization, you don't have to work. You can prepare all the materials.
Yeah, the animal's doing the work for you.
Do you see any benefit to going past 5 weeks? I mean, if you stop at 5, is it different than stopping at 10, or is it, your accelerated immunization gives you things just as much diversity?
It will be pretty much the same for most times. We had
for most things
for snake phenom. There we had to boost again, so we couldn't get good binders out of it, and then so we reboosted the animal, but that was 3 months later, and then it gave a much higher titer. But that might be because you start with snake phenom, you start with extremely low doses because you don't want to kill your animal. So you start with extremely low dose and you go up. So that might have been the reason. While normally you start with a high dose and then you go lower in dose, yes, for the immunization to stimulate the affinity maturation. And by the way, because very often I'm hearing that they say, yeah, but you need much more antigen because you have to immunize. We were immunizing standardly with 100 microgram per shot. And actually, because the affinity maturation and the proliferation is going on in the animal, then the library has to be smaller, so it's easy to generate. And also the panning is very easy compared to those large synthetic libraries. And I have knowledge of people, including ourselves, we work with a synthetic library. You have many more binders, but then you have to screen all those binders to find out which is the best one. And very often you have the K-on and K-off rates are jumping on it and falling off immediately. So you have to remodify your best binder to try to have the kinetics better. And then, so you need to make a second library on your first binder, and then you need to screen it again. And so it takes more time and more material. It's best just put it into the animal. And by the way, we preferred in the beginning camels or dromedaries because in those animals you have about 50% of their antibodies are heavy chain antibodies. In llamas, alpacas, it's only 25%. So this suggests that it's more important for those animals and that you will have a larger diversity. You will have more binders from a dromedary. But from immune libraries, so most of the time we only retrieve between 5 to 10 binders, because you're more stringent in the washings so that the weak binders, you get rid of it. They are eliminated.
I guess on the topic of the different isotypes, and a lot of people that we talk to are surprised to hear that camelids make both conventional antibodies and heavy chain only, and that they occur in different ratios. I guess, have you seen any bias in the response that, you want everybody who immunizes a llama, alpaca, or camel wants the heavy chain only, but sometimes you get a much stronger IgG1 response. I guess, have you seen any kind of rhyme or reason to that in terms of antigens or immunization strategy?
I fully agree. Sometimes we see also that the immune response in the IgG1, the classical antibodies, is higher, much higher than in the heavy chain antibodies. But I can't tell why. But listen, those are outbred animals, yes? So if you're not sure, just inject it in two animals. Of course, it doubles the price for the immunization, right? So we at the university, what we did the opposite. I know Ablings, they were immunizing, well, I don't know, 5 llamas or alpacas with the same antigen to have more binders, yes? So they made 5 libraries and 5 pannings and so on to have more diversity to choose the best one. We put 5 to 10 antigens in one mixture and we give it simultaneously to the animals. And then by panning, we sort it out. But it normally works every time. It's 95% success rate in our hands.
That's interesting to hear you say that, because we actually just started we just started a project last year where we started multi-immunizing with different targets in the same animal. And we were expecting to see a lower response per antigen, thinking the immune response has to split itself, but let's try it anyway, because we get vaccinated against lots of things and your immune system is your immune system. And it worked great. We saw a very nice titer to all of the antigens. However, it was different in terms of how much canonical versus the heavy chain only. Those were different. And there was, again, no rhyme or reason to it. I'd love to understand. I think a lot of people would love to understand how to supplement a G2/3 response versus a G1 response. But we've had the same where we have multiple animals on one immunogen, one animal on one immunogen, one animal on multiple immunogens. It doesn't matter. It's kind of a pick and choose.
It goes in all directions.
But the longer, so we will, for some of our animals, we'll inject multiple times over multiple years because we're making polyclonals for some of these things, polyclonal VHHs. And we have seen, surprisingly, after about 9 months to a year, your heavy chain response, instead of, I think you cited 25% or so in alpacas, you'll see a big increase after about 9 months to a year, a big increase in the G2, G3 response. So if you're using one animal and you come back a few months later. It also, we saw the same thing with multiple injections that helps. And sometimes the amount of the immunogen, once in a while, we've seen projects where that helps, where you increase the amount of immunogen and the frequency of immunization. And if it's an animal that's been immunized for a year or more, you see a massive titer response in the G2, G3 versus somebody who's been injected for just a few weeks.
See increases in viral neutralization after several months. And so if we're going for neutralizing titers, that, especially in alpacas, takes longer to mature. One of the things that we've started doing to pull things out is using alternative adjuvant systems in the late boosts, where it's moving away from CFA, IFA, and towards different TLR pathways. My theory is that maybe there is something there where we could sort of engineer an adjuvant strategy to tease out additional 2+3 responses, especially in later stage injection boosts.
Yeah, I think there's something there too. I think the biology is so fascinating. And there has to be something to the effector functions of these subclasses, just like humans, G1, 2, 3, 4, they have different effector functions and they trigger the immune system different ways through those FCs. So there must be a correlation there with camelids. And I've been watching the literature avidly to see if somebody figures this out. And I have yet to see anybody figure it out. But I agree. If that's the case, if it's an effector function, then you could certainly add an adjuvant to supplement that effector function for that part of the immune system. But it's still a mystery right now.
Absolutely.
I don't know which adjuvant you're using. We were using always an adjuvant, Gerbu adjuvant, which is used for cattle, for farm animals. Yes, that's the one we used. I do know originally we were using complete Freund, and then later on for the boost, incomplete Freund's adjuvant. The problem is that the animals get ulcers from it, certainly if you give it repeatedly. So that's why we avoided it and we switched to the Gerbu adjuvants. Now, there is one other aspect that I would like to mention, that is that in the genome, you have the VH germline and the VHH germline. They are in the same cluster. So they have to rearrange with an IgM first. Yes, and when you are rearranging a VHH, it has to go to the Ig2 or 3 subclasses. Yes. If it's a VH, normally it goes to the Ig1 subclass. You have Ig1A and B, right? However, there is a problem sometimes during the recombination, you have a deletion and you obtain a VH domain that is not fit to bind to a VL. And therefore, those ones, they are also going to end up in the Ig2 or Ig3. So sometimes if you make your library on heavy chain antibodies, on the VHH, you have in there a number, it can be 10, 15% of them, they have a VH imprint in their framework too. But you might say, well, this is not what we want. And to be honest, we discarded those in the very beginning because we said that this is contamination, so we don't want to work with it. But later we found out that they have very often the framework 4, there are amino, and the tryptophan has been removed by deletion. And that's important for the VL contact. And so those ones, we call them VH-like nanobodies, those are very good because they are nearly human. Also, the framework 2 is like human. It's exactly the same amino acid as humans.
So you brought up the IgMs, and I guess one maybe silly question that I've always wondered is when heavy chain onlys go, when they're IgM stage, do they have a light chain? Is it just like a surrogate light chain that's there? They're single chain IgMs with no.
Yeah. Well, no, actually, we don't know. Before the light chain is put on it, you have expression as a B cell receptor, an immature B cell receptor, with the surrogate light chains. And the surrogate light chains, you also have a VL domain and a CL domain. But normally there are two different polypeptides. But the V pre B, as it's called, cannot bind to the VHH domain. And therefore, those B cells that have rearranged a VHH, yes, they can, if this happens in our system, in human or mouse, those B cells are going to end up in apoptosis. They will not survive. But the camel has an escape route. The escape route is link it to the Ig2 or 3, and then you can have maturation. Because if you do messenger RNA sequencing of the IgMs, you will find with a VHH, they have the CH1 domain, but they have no mutations. They're completely virgin VHH germline. Yes. But when it is on an IgG, you have several somatic mutations in there. So
I always wondered that.
it's a transient expression on the surface of the B cell probably, but then it gets, or perhaps not on the surface because it cannot go through the ER. Yes, and then they have to class switch.
Got it. I guess, okay. So then kind of jumping back to other questions that the community has asked, a lot of people have concerns about affinity. So because you're only engaging with three different CDRs, you're only engaging the antigen with three CDRs. Is there a fundamental limit to affinity? Is there kind of, do you have to affinity mature after to get a, nanomolar or subnanomolar binders? How do you guys see that happening in your campaigns?
I can speak for myself first, and then I'll let Serge add his thoughts. We've gotten, double-digit picomolar straight out of animals before any maturation. It's not particularly rare. And then I think the sort of obsession with affinity for certain applications is starting to be sort of teased apart, especially in therapeutics. So if you're looking for long-term inhibition, then yes, it's absolutely important. If you're looking for payload targeting, absolutely important. But in these multi-specific applications, especially where there's avidity involved, single-digit nanomolar is really all you need in many cases.
Yeah, I agree. I think the avidity part is a big component there when you're talking the multi-specifics. And we saw the same thing. You can get subnanomolar binders right out of the animal. You just have to have the right screen for them.
Yeah, it's true. Well, there is some confusion because most people compare the affinity of an antibody, monoclonal antibody, with a VHH. And then, of course, you have a bivalent construct against a monovalent construct. So we are normally losing. But if you convert the nanobody into, again, a heavy chain antibody construct, or as a dimer, a tandem repeat, you increase avidity effects and well, you obtain avidity effects, and you will have a higher functional affinity. So they're normally, listen, you just think of it. If you're immunizing the animal, in the animal you have the classical antibodies and you have the heavy chain antibodies. The fact that those are co-maturing means that the affinity of the heavy chain antibody is as good as the classical antibody. Otherwise, they could never compete. Why should you raise an immune response if it's never used? Why should you keep it? You will not keep it because the animals have it, so it gives them an advantage. The affinity should be at least workable. It should be practical point.
So then I guess looking kind of VHH adjacent, have you worked at all with IGNARs? Anybody? The shark single chain antibodies?
We've looked at them. My understanding is that without sort of grafting approaches, they're very difficult to take to a humanized form. I know there have been a couple sort of grafting approaches onto a humanized scaffold, and it seems like in certain instances, some frameworks will graft pretty well and it's well tolerated. And I think that is a potential sort of discovery library path forward if you're doing a synthetic approach. But at that point, to me, it seems that you're going to really have a workflow that becomes difficult to maintain and scale. And at some point, you'll probably move to a synthetic workflow anyway.
Yeah.
I'm not so sure about humanization. First of all, we did a test whether it was immunogenic, so with human immune cells, T cells, dendritic cells, etc. The uptake, the expression, well, the exposure, and so on. We couldn't see any difference. Yes, so it was not immunogenic. By all means, it was not immunogenic. But people, if you want to commercialize it for therapy, of course, for the perception, you will say, well, we are going to humanize it. Now, the humanization of an antibody is pretty easy, because what you're going to do, if you want to use it for therapy, you would like to have the highest possible expression. So you will go to Pichia or yeast or Corinex. It's a Japanese company that is using gram-positive bacteria that gives you high yields. Okay, and secrete immediately in the medium. So what you want to do is you want to use your nanobody sequence and to convert it to the codon usage of the host in which you're going to express it. Right? So you're going to synthesize the gene again. And during that synthesis, you just those 2, 3, and well, 4, 5 amino acids that are different between a VHH and the human VH, you just change them. I do know at the antipode, so on the opposite side of the paratope, you have in a turn, you have a proline, and in human it's an alanine. And in another turn, it's an alanine instead of a proline. You just switch them. It's exactly the same. It doesn't change. And also there are a few mutations, an arginine, a lysine that has been substituted. So before doing it, what you can do, we have about 20 VH of family 3 in human. Yes, you take those and you align those VH germline, human VH germline, and you align it to your alpaca sequence, right? And you see where the differences are, you see which is the closest to which human gene it's the closest, and you change it to that one. Except for framework 2, there you have to be careful, because if you have a long CDR3 loop, it falls over that region, and if you change the framework 2 regions, you might change the conformation of your CDR3 loop. So there you have to be careful. But for the others, it's piece of cake.
If you, in terms of like amino acid sequence humanization, true, but functionally speaking, I don't know that you really need to do that. And I will preface this by saying we don't make therapeutics, and that's not the target of the things we're making. We're making tools. But we make anti-VHH, and the VHH that we use for immunogen, then, you know, the anti-VHH that we get out of the animal, we have the hardest time cross-absorbing against humans, because we want to have anti-VHH that's minimal cross to human IgG, right? So to have it recognize alpaca VHH or llama VHH and not recognize human VH is extremely hard to make. And we have so low yields at the end because they're so similar. The human and the camelid VHH is just, you know, so homologous that functionally speaking, they look alike to an animal like a goat. So for, you know, to put it into a human, I would assume similarly, it's going to look like human. So it's not going to cause an immune response necessarily, but.
There is one exception. So there are people, so in those clinical trials, they have noticed that according to one report, I think 25% of the humans have antibodies against VH. Yes, VH or VHH. And then it's again against what I call the antipode, and the opposite of the paratope. And so Ablings, because they have patented it, and they notice that when you extend the C-terminal end with 3 alanines, you avoid that those antibodies binding to your VHH. And the reason they found it's just a cloning artifact, because after the VHH, we had the NOT1 restriction enzyme site, and we were putting it in the frame so that it generates 3 alanines. And then they put a stop codon, the amper stop codon. Well, I do remember this because we were laughing at it. They say, yes, the camels, and you are like an Arab, so you should have known it should be in the Allah reading frame. So that's why I remember it. So if you keep those 3 alanines, you don't have the issue anymore. according to that patent.
And this is where the bispecific linker from the Ablinks molecules comes from, I assume.
Yes,
Because it's
yes.
an AAA as the bispecific linker.
Yes. Yeah.
So that's a good question because a lot of the companies that are interested in developing VHHs are interested in developing multi-specific platforms. And their concern is how humanizable is this different construct? So yes, if you just kind of look at a VHH versus a VH, you can humanize it. But then if you have a daisy chain, or if you have something off the FC, or if you have this mono FC, like how does that play into humanization? Is that still doable?
What we were using for that reason was we were using the IgA linker of the human IgA linker. It's something with a lot of proline, so it keeps the two domains separate. So you have your VHH always at the N-terminal end, IgA hinge, and then your second part, whether it's a GFP or whatever it is. Then you also avoid immunogenicity in humans.
That's clever.
It's not clever, it's chemistry.
So then I guess because this is the year of AI generated drugs, and everyone's talking about AI generated antibodies, and VHHs are maybe the lowest hanging fruit. I guess based on what you've seen, do you think that we're near the next VHH that's been completely in silico designed in the clinic? Are we putting llamas and alpacas out of jobs?
I think that for ideal scaffolds, that we start from structure and we're in-painting CDRs against known antigen structures, I think we're probably pretty close. I think for true de novo generation, where we are engineering frameworks as well, and especially against unknown structures, we're still 10 years out. But I think that there's always going to be a place for immune repertoire in sort of the bigger picture.
Yeah. But on the other hand, I think it's a very clever idea to try. So whatever protein you have, whatever target you have, with AlphaFold, you can predict the structure. It's quite accurate in most cases. Yes. So, and then once you have the structure, you can say, well, I want antibodies on this epitope. This should be the epitope, right? And what you can do, there are now 800 to 1,000 different VHHs in the structural database. So you take those 800 nanobodies, the antigen binding loops, right? All the amino acids that are facing inwards to support the loop, you keep them. The ones that are sticking out, reaching into the solution, those you put in silico to a glycine. And then with that scaffold, those scaffolds, you see which one is the best to fit on your epitope, right? And then once that's done, if you see the C alpha fold, this best fitting, then you start to generate the side chains again. So whenever you have on the antigen an arginine, you put a glutamic acid or an aspartic acid to have the charge chain. Complementarity, you have van der Waals interactions, you try to fit it as much as possible. That's the way to proceed. So what will be the affinity that you get? I don't know. It might be like David Barker has done, so it might be something like 50 to 100 nanomolar. It's very close. And then you could do also some randomizations and do a panning again to have a better fit. The advantage is that you will bind to the wanted target, the wanted epitope. Yes. So not if you bind on the other side, it's not interfering with receptor binding or ligand binding. So those are useless, those nanobodies. But the one that is binding to your target epitope, that's very important. And so that will work. That will work. But how long will it take before you get it into the clinic? That's another question. Yes. That's another question. That will take, if it's not SARS-CoV-2, it will take you another 10 years. Huh? I would like to come back on one thing of the steering of the immune response towards the heavy chain antibodies. You can do this with those xenomice that are available now. That are at least made, that have been made. Yes, in the xenomice, what they have done is they removed the CH1 of the endogenous IgM and IgG, And so therefore it can only produce heavy chain antibodies. So in that case, you don't have the problem of competition with classical antibodies. So there you immediately obtain heavy chain antibodies only. Yes. So no competition with.
Question on the sort of topic of mouse models. I've always wondered if you are actually getting CDR3 length sampling from the mouse maturation machinery that is similar to what you would find in a llama or alpaca or camel. One of the sort of values of the animal maturation is this ultra long CDR3 sampling. And so I'm curious to hear if everyone thinks that heavy chain mice are within a few years of getting there, or is there sort of more work to be done and that's a big gap?
I think personally that the on average longer CDR3 length, especially in camels and dromedaries, it's part of the selection. The ones with the shorter, they have not been selected because they cannot bind as good. Yes, so you have a selection towards. Oh, by the way, in llamas and alpacas, we do have VHHs with a short CDR3 loop. But most of the time standing up, right? And so it's binding from its side, not from the top. We have some crystal structures of that type. But I think it's most of a selection that you have. And so that will happen in the mouse as well, I suppose. It will generate different, although it might be that the D genes in mouse are shorter compared to llama. And therefore, on average, the CDR3 is even shorter than human. But they have xenomouse with a D diversity, the D genes from the camel or the llama. So it would change there.
Do you think that's why we struggle sometimes to find heavy chain only antibody binders to small targets like peptides or small molecules? It's because they're generating short CDR3s and then they're being outcompeted by the conventional. Is that why we see conventional responses to these smaller targets, do you think?
The reason is that, yeah, that's a good point. The thing is that if we immunize with proteins, you always have, even small proteins, you always have good binders. If you immunize with a peptide or a hapten, it's difficult to have good binders. The reason is that in a conventional antibody, you have your VH and VL, and they can form a groove or a cavity that where the hapten or the peptide can fit. On a VHH, it's like an egg shape, yes? And so, yes, that there is no cavity, there's no groove. You have some examples. We have a few binders, against peptides. And there you see that those loops are opening a little bit so that it can fit, well, within the CDR loops. But it is difficult. It is difficult. We have much lower affinity, at least in our hands. When people came to me and they said, we have a peptide or we want to immunize with a peptide, I was charging more. Let me put.
We've experienced similar with haptens, with peptides, and also with intrinsically disordered proteins. And
Yes,
so I think there's some secondary structure impact on how the VHH will interact.
Yeah,
Certainly.
but that's exactly the same, huh? Intrinsic disordered protein. So it has a high
The fancy name for a
value, and
peptide.
you're going to fix it in one conformation. So you have an entropic penalty of binding. Yes, you have
Exactly.
to overcome it with enthalpy. And yeah.
Yeah, we've seen the same. If you immunize with a peptide, we'll see a nice titer in IgG1. But it doesn't matter. You can immunize for 9 months to a year. We still aren't seeing titer to the G2, G3. We'll see G2, G3 response to the carrier, like ovalbumin, but we won't see G2, G3 response to the, not significant anyway, to the peptide, unfortunately. So we've been struggling with the same thing because that's a tool that the field would use. I think that's going to be difficult to get there just because of the biology of the VHH itself, I think.
Mm-hmm.
This has been a great discussion. A lot of questions I've had in my mind. I guess we all have the same questions, but
I think the gaps are common for this field. Absolutely. And it's still young, and I'm excited to see where it goes.
I'm especially happy if I see that in Europe and in US, they have the same problems and the same solution for using the antibodies. So.
Yes, well, I thank you all so much for your time. And I'll definitely let you know when the resource becomes available. This is going to be, I think, very useful for the community. So thank you again.